swarming nanomedicine and how we use swarms of robots to help us design, hopefully, next generation drugs, and how that can help us make robots do things like clinical skills, hopefully, in the future. So I'm fascinated by these swarms. If you look at flocks of birds, they can do these beautiful, complex behaviors. And there are many features that are interesting for real-world applications. So for example, if you keep adding birds to the flock is still going to fly, so we can scale to large numbers. If a bird falls from the sky, the whole flock doesn't fall to the ground, so they're very robust. And together they can do more than the individual birds. For example, they're better at avoiding predators. And so the question is, how can you take these ideas from swarming and put them into the real world? And so this was the subject of my PhD over in Switzerland, and we could really just throw 10 of these flying robots. I'm behind the camera here. And using basic rules, you could have them do things like flocking behaviors. So here you see 10 of them in the sky. Sometimes a bird comes along and follows them because they're about the same size. And these are the GPS trajectories. So it starts out very messy, but over time you get a circle. There it is. And that's actually what we had predicted using our simulations. So in the world of swarming, we can now take basic rules, simulate them to understand what the group does as a whole, and then hopefully push these out into the real world. So that was fun, but that was 10 robots, and I always thought that swarms needed to work in the hundreds and the thousands and in the millions. And so I got excited about the field of nanomedicine. And nanoparticles think of them as tiny spheres that are good at basically leaking down into tumor environments because of their size. And so that makes them interesting vehicles to deliver a drug or to deliver a treatment to the tumor environment. And it turns out that these nanoparticles work in the 10 to the power 13. So that was the very large number that I was interested in. Obviously, being able to help people with cancer is a nice application of this. And the laboratory where I went to for 3 years over in Boston made nanoparticles for cancer treatment. And they came in lots of different flavors, so you could change their size, their shape, you could change their charge, and change what they stick to. You could decorate, well actually you could change their material, so you could use magnetic fields or light to activate these particles. You could decorate them with molecules, and that allowed them to bind to molecules on the cancer cells or to bind to each other. And you could load drugs in these particles that you could release in a more or less controlled fashion. And so there's lots of knobs that you can turn on the design of these nanoparticles. And very often I get the question, well, that's great, but how would you make these nanoparticles swarm? Well, the birds had a brain, and that was probably telling them how to behave. And our flying robots had a program that I put in them. And in nanoparticles, you don't have a brain or a program because they're too small and simple. But what you can do is figure out how you turn the knob on the design of these particles so that when you inject them in the system, they behave as a collective in the way you want them to behave. And the reason we're interested in doing that is really all these words that we take from the swarm robotics literature. So can you have particles that synchronize, that create trails a little bit like ants to your picnic table, that make decisions about the state of a tumor? And if we could do that, could that be helpful for clinical applications? This is not a completely crazy idea. So the laboratory where I did my postdoc designed a system and tested it in vivo, so in living systems, which had two particles. First, they'd send in these blue particles. They'd arrive through the bloodstream, leak out into the tumor tissue, and these blue particles were made out of gold. So if you shine near-infrared light on them, basically a little laser, they vibrate, heat up, cause damage to the tumor tissue. And then they send in these pink particles, arrive through the bloodstream, but these pink particles have molecules that bind to the damage that was just created in that tumor. And you get 40 times more drug delivered to the tumor using that system than if they weren't communicating. Somehow they're communicating through the tumor because one did something to the tumor that the other can sense. So that's the big picture, but we did want to dive into something very concrete that can be helpful for bioengineers now. And there's a little bit about this modeling, but it's going to go back to robotics afterwards. And we asked a simple question. We said, what if you have a vessel and you want to go into kill 20 cells deep from that vessel? How should you design your particle? Because remember, that's how we're controlling them, in terms of how big that particle is and how sticky that particle is to the cancer cells. And so if you do simulations of this, and we've got lots of numbers that are realistic for the cancer field, this is what it gives us. And there's really just two axes here: how fast it is, that's the size, how sticky it is. And our goal is to kill 20 cells, that's the dark red. And the first thing we realized is most of these particles don't kill the 20 cells, so it's a hard problem. And then you can dive in and look into these boxes. So if I take A, for example, A was a particle that was very sticky. And what happened is it leaked out of the vessels, and the first cell ate up all the particles. And so they never went deep into the tumor tissue. And that was a little counterintuitive for the bioengineers, because when they're designing their particle, they want it to be really sticky, because then it gets eaten up by cancer cells. And so it's interesting to think of collectives and dynamics rather than the individuals, and this was a proof of concept. And then you can come up with rules that are easier for them to use. So we said, why don't we prevent these particles from sticking, so we have a shell on them, and we let them go deep into the tumor tissue, take that shell off, and then they can finally stick to the cancer cells and get eaten up by them. And it turns out that if you do that, you can solve for all the stickinesses, for all the sizes, the killing 20 cell problem. And there are actually particles that do this. So this is a nanoparticle that was just a couple floors up from where we were that had this shell mechanism that could take off that shell, and they showed that it goes deeper into tumors. So that was great, but that was just one problem, spending 2 years thinking about nanomedicine. And so what we did is that we designed a game so that we could explore many more scenarios and have the crowd come in and help us design these nanoparticles, because there's 10 to the power 22 combinations of particles that you can make, and that's tricky computation we need to go over. So essentially those are our scenarios, and then the crowd is on nanodoc.org if you want to play, can design these particles for us, and when they click inject, that goes back to our scientific simulator. So this is what we teach the crowd. We teach them about cancer cells, we teach them about healthy cells. You know why we need healthy cells in our game? Essentially the solution to everything is to dump loads of drug on it, and that's obviously not what you would want in a human. So having a healthy cells makes sure that people have treatments that are specific. You can change the dosage when you inject them. You can combine them, so those pink and blue particles are something I'm interested in. Change their size, that changes their speed. Some of these cells have receptors on their surface, and you can make particles that have a similar surface, so they're going to stick to those cells. You can load them with a drug, and you can have smart materials, where if something binds to that particle, it's going to release something. And then if you're a roboticist, you start thinking, wait a minute, now I have a sensor, I've got an actuator, so I can send something out into the environment to be able to communicate potentially with neighboring particles. And the last thing that was missing was the ability to move them in different ways. You know, my flying robots, they could go left or right or straight based on what I told them. But you can't really do that in the particle world because things move randomly. What you can do is self-assembly. So particles can come together and come apart, and that slows them down or speeds them up. So that's how we're controlling motion to a certain extent. So we've had around, actually now it's 150,000 simulations run by the crowd, and we're currently going through that. And this is just a snapshot of one of the things that the crowd came up with. And I'm not going to go into detail because I want to put us on track again. But these are the things that come out of the simulations, and they're quite counterintuitive. And this one was fun because the solution was to create two particles, one which sent a message to the other one. And actually that's something that we had never taught the crowd to do. So they can think outside of the box, and we can really just throw problems at them and see what innovative solutions they come up with. And actually in our lab we play our own game to go back to this idea of swarming. So this is an example where you have a vessel and a rare event, some mutation or stem cell, and you get these ant-like trails between those 2 points. And we actually started from the ant rules and simplified. And it turns out you can do something similar in simulation with nanoparticles. So that's all simulation, but like with the flying robots, we want to be able to put these things outdoors. And so the lab and I built 1,000 little kilowatt robots, which you're going to see at noon in the robotics lab. And these were originally designed by Roderick Nagplas lab over at Harvard, and she nicely gave us the designs for these. So we have 1,000 of them. They can vibrate, they can talk to their neighbors, they can sense their environments. And this is just what it looks like when we run the nanoparticle rules on these robots. So here in red you have cancer cell robots. In green you have nanoparticle robots who are leaking out of this artificial vessel. And then when they stick, they turn blue. So this was a case where the particles were too sticky and they never go deep into the tumor tissue. And this is just a really powerful way to visualize dynamics. In just 10 minutes you can put this on a robot swarm and see how these behave in the physical world. And that removes some of the assumptions that we had in simulation previously. And it's just a good tool to think differently about the problem. So this is the 1,000 robots. Actually, they're not all in the picture, but what we're doing now is that we can project these tumor environments on our arena, and we spent a lot of time figuring out, if you have a nanoparticle, and blow that up to a 3-centimeter robot, that's a scaling factor. And so you can do that for the cells, you can do that for time, and actually the things we're running on the robot swarm are proportionally at scale to the nanobots. The things we're visualizing make some sense. But ultimately, the robots are not the validation. What we do is do these at the nanoscale with nanoparticles so that we can see them under the microscope. And we build these little microfluidic channels using 3D printing so that we can see these dynamics. So this is what it looks like in our channels. The red are cell-like structures, the green are our particles, and we're currently working towards being able to functionalize this so that it's more tumor-like. And because we have these channels, we can also play around with other robots that can be more controllable. So here what you're going to see is work with a collaboration with ETH Zurich. And we have a little micro robot, which is really micron size, so the size of a cell. It's a magnetic coil, and if you turn magnetic fields around it, this coil spins. And so what we're doing here, everything that's bright is a nanoparticle. And remember, these are hard to control. But that thing that moves in there, can you see it? It's a remote-controlled micro-robot. And those actually can propulse these particles and do things that are interesting. For example, pushing them into tumor tissue. So we're just exploring ways of combining the many nanoparticles with maybe smaller and more controllable robotic agents. So that's the general idea. We try to go from what's the swarm behavior you want to figuring out, well, what does it take to design that swarm behavior, testing that on robots, testing that in microfluidics, and we work a lot with clinical collaborators, for example, here in Bristol for brain cancer, to see if we can come up with guidelines that would be helpful for them. And really back to the robotics world, I think that as we simplify our rules to make swarms of trillions of things work together, that's going to provide valuable insight for robots that we want to deploy in oil spills or in forests that also work in the thousands, because we're not going to be calibrating all of them. They're not going to be reliable because they're cheap. And so trying to figure out really what's the basics that we need to deploy this can be helpful. So here what you're seeing are 100 of our robots, and they're going to do this ant-based trail formation, but as if they were particles. So it's so simple to put this on there. And so here they look very organic, so you can kind of see them dance around each other. And over time, they create a trail to that robot at the upper right. And this is something where it didn't take any fine-tuning. It's just such a basic, simple set of rules that you put it out there, and there's your trail. All right. So thank you. I'll take one question, and then we can move along.
Thank you. It's very interesting. I have a question regarding the time to market of those technologies. I'm not talking about the nanomedicine because it will take time for clinical trials and the introduction to the market, especially in Europe, will be challenging. But clearly swarming is today some kind of route of the robotic industry and in many applications. My question is really how close to the market are we today on specific, let's say, inspection or surveillance application using drones or remote operating system on the ground? Do you see today fields? I mean, we have the need, clearly a need of using this kind of technology, but do you see some solutions today that could make an impact in the short term, or are we still talking about a very, far from the market research perspective?
Yeah, so two parts. I think in terms of the clinical applications of nanomedicine, some of the guidelines that we're issuing are for drugs that are nearly FDA approved or FDA approved in the US. So those are actually closer to market than the swarming nanomedicine, I agree, is going to take way more time. In terms of the swarm robotics, for the 10 flying robots, to get them in the air, we did not have any pilots or backup pilots with this. So that means that we had to put in place something that's extremely robust and fail-proof in terms of we knew they weren't going to fly away, we knew that GPS crash, they would do something smart. And I think because we had to go through those steps of getting 10 in the air, that actually puts you in a good position for commercializing the swarm approach because you've thought about all that's needed to make your system very robust and reliable. So I think those lessons have the potential to translate to systems and robotics for commercial applications with low number of robots. So, the sub 50 or so. I think the thousands is really where it starts to be interesting for a lot of things like oil spills. It wouldn't make sense to send out potentially hundreds of these robots out there. And I think we're just learning now how to control very large numbers.